Hello everyone. Welcome to BSc Biotechnology third year. Today we will be starting with Biotechnology paper 1, which is Molecular Biology and Genetic Engineering. We will start with the first unit, and the first topic of the unit is Introduction to Molecular Biology. I am Mrs. Pragya Goyal, Assistant Professor, Department of Biotechnology, IPS Academy, Indore. What is molecular biology? Molecular biology is the field of biology that studies the composition, structure, and interactions of cellular molecules, such as nucleic acids and proteins, that carry out the biological processes essential for the cell's function and maintenance. While traditional biology concentrates on studying whole living organisms and how they interact within populations, which is a top-down approach, molecular biology strives to understand living things by examining the components that make them up, which is a bottom-up approach. Both approaches to biology are equally valid, although improvements to technology have permitted scientists to concentrate more on the molecules of life in recent years. When we study any science, we cannot study it in isolation. Each science, specifically biology, is related to different fields in biological sciences. So when we talk about molecular biology, it is related with biochemistry and genetics. Molecular biology, as we just heard, is the study of molecular understanding of processes of replication, transcription, translation, and cell function at molecular levels. What is biochemistry? It is the study of chemical substances and vital processes occurring in living organisms. Biochemists focus heavily on the role, function, and structure of biomolecules such as proteins, lipids, carbohydrates, and nucleic acids. Molecular biology is also related with genetics, which is the study of how genetic differences affect organisms. Genetics attempts to predict how mutations, individual genes, and genetic interactions can affect the expression of a phenotype. While researchers practice techniques specific to molecular biology, it is common to combine techniques in molecular biology with methods from genetics and biochemistry. Recently, a significant amount of work has been done using computer science techniques such as bioinformatics and computational biology. Molecular genetics, the study of gene structure and function, has been amongst the most prominent subfields of molecular biology since the early 2000s. The molecules which form the basis of life provide scientists with a more predictable and mechanistic tool for scientists to study. Molecular biology provides scientists with a technique with which they may understand the way life works. They may use them to determine the function of single genes or proteins and find out what would happen if that gene or protein was absent or faulty. Molecular biology is used to examine when and why certain genes are switched on or off. An understanding of each of the factors has granted scientists a deeper understanding of how living things work and used this knowledge to develop treatments when living things don't work so well. In this paper, we will be dealing with fundamental processes of life like replication, gene expression and its regulation. We will also talk about classical genetics and evolution of life. Further on, we will proceed to genetic engineering in Unit 4 and Unit 5. To start off, there are so many terms that we use in molecular biology, but not all of them can be covered here. So here we are going to talk about few of the fundamental terms which form the foundation of molecular biology, for which the understanding is very necessary before we can proceed to higher topics. The first term is nucleic acid. Nucleic acid are the biopolymers or small biomolecules essential to all known forms of life. The term nucleic acid is the overall name for DNA and RNA. They are composed of nucleotides, which are monomers made of three components, that is the nitrogenous base, sugar, and a phosphate. The next term is DNA. Deoxyribonucleic acid, which is the full form of DNA, is a molecule composed of two polynucleotide chains that coil around each other to form a double helix carrying genetic instructions for the development, functioning, growth, and reproduction of all known organisms and many viruses. DNA bears the hereditary information that's passed on from parents to children, providing instructions for how and when to make the many proteins needed to build and maintain functioning cells, tissues, and organisms. So, how does DNA carry this information? How is it put into action by cells and organisms? This is a very complex, fascinating, and fairly mind-blowing process, and we will explore it in more detail in this paper. Next term is ribonucleic acid. Ribonucleic acid or RNA is a polymeric molecule essential in various biological roles in coding, decoding, regulation, and expression of genes. Just like in DNA, RNA is made of monomers called nucleotides. It is principally involved in the synthesis of proteins, carrying of messenger instructions from DNA. In some viruses, RNA rather than DNA carries genetic information. It is of three types: mRNA, rRNA, and tRNA. So as we can see from the previous diagram, DNA is a double-stranded helix, while RNA is a single-stranded molecule consisting of essentially the same monomers, that is nucleotides. The next term is the gene, which is a very important term. Gene is the basic physical and functional unit of heredity. Genes are a distinct sequence of nucleotides forming part of a chromosome. Some genes act as instructions to make molecules called proteins. However, many genes do not code for proteins. Instead, they code for RNA. The order of nucleotides determine the order of monomers in a polypeptide or nucleic acid molecule which a cell or virus may synthesize. In humans, genes vary in size from a few hundred DNA bases to more than 2 million bases. The next term is the exon. Once you have understood what is a gene, we need to understand which parts are the coding sequences and which parts are the non-coding sequences. Exon is a coding region of a gene that contains the information required to encode a protein. The term exon refers to both the DNA sequence within a gene and to the corresponding sequence in RNA. That means exon is found in DNA as well as in RNA after the process of transcription. Moving on, the non-coding part of the gene, which is the intron. It is a long stretch of non-coding DNA found between exons or coding regions in a gene. They are parts of the gene sequence that are not expressed in the protein. They are called introns because they come in between or interfere with the exons. Genes that contain introns are known as discontinuous or split genes, as the coding regions are not continuous. Introns are found only in eukaryotic organisms. This means the whole of the prokaryotic genome is the coding part. They do not have introns and hence they do not have non-coding portions. The next term, which is again an important term, is the term genome. A genome is the genetic material of an organism. It consists of DNA or RNA and RNA viruses. The genome includes both the genes, the coding regions, and the non-coding DNA, as well as the mitochondrial DNA and chloroplast DNA. As we all know, these are the two semi-autonomous organelles which contain DNA. They also form part of the genome. The study of the genome is called genomics. Each genome contains all of the information needed to build and maintain that organism. Simply put, it is the haploid set of chromosomes in a gamete or microorganism or in each cell of a multicellular organism. The human genome is a complete set of nucleic acid sequences for humans, encoded as DNA within the 23 chromosome pairs in cell nuclei and in a small DNA molecule found within individual mitochondria. These are usually treated separately as the nuclear genome and the mitochondrial genome. In humans, a copy of the entire genome, which is around 3.2 billion base pairs, is contained in all cells that have a nucleus. Just a thought, what would happen if we printed out the 3.2 billion letters in your genome? What would we get? We would get a stack of paperback books 200 feet high, which is equivalent to 61 meters. We would also fill directories of 500 page. We will fill 200 directories of 500 page each. Or suppose if we recited at 1 letter per second for 24 hours a day, it would take a century, 100 years to recite the complete genome, human genome. The next term is the chromosome. In the nucleus of each cell, the DNA molecule is packaged into thread-like structures called chromosomes. Each chromosome is made of protein and a single molecule of DNA. Passed from parents to offspring, DNA contains the specific instructions that make each type of living creature unique. The term chromosome comes from the Greek word for color, that is chroma, and body, that is soma. Scientists gave this name to chromosomes because they are cell structures or bodies that are strongly stained by some colorful dyes used in research. When the cell is not dividing, chromosomes are not visible in the cell's nucleus, not even under a microscope. However, the DNA that makes up chromosomes become more tightly packed during cell division and is then visible under a microscope. Quite fascinating, isn't it? How so much information is packed into a chromosome, and this haploid set of chromosome forms the genome. I think you can understand the importance of the information that is there, because it is passed on from one generation to the next in the way as it was, without much of a change. Moving on, next term is diploid. There are basically two types of cells in the body, haploid cells and diploid cells. The difference between haploid and diploid cells is related to the number of chromosomes that the cells contain. Diploid cells have two homologous copies of each chromosome, usually one from the mother and one from the father. This number is depicted as 2n. In humans, this number is 46. The next term is the haploid. Haploid describes a cell that contains a single set or half the number of chromosomes as compared to diploid cells. The term haploid can also refer to the number of chromosomes in egg or sperm cells, which are also called gametes. In humans, gametes are haploid cells that contain 23 chromosomes, each of which have one of a chromosome pair that exists in diploid cells. The number of chromosomes in a single set is represented as n. This is known as the haploid number, and as just mentioned, this number in humans n equals to 23. Now we come to another term, monoploid. If we look at it, mono means one, ploidy means set of chromosomes. So here we are also talking about one set of chromosome. In haploid also we are talking about one set of chromosome. But there is a difference between the two. In case of monoploids, there is evidence that n chromosomes in a haploid set have resulted from duplications of an originally smaller set of chromosomes. This base number, the number of apparently originally unique chromosomes in a haploid set, is called the monoploid number. It is also known as the basic or cardinal number or fundamental number. A very good and a very common example to understand monoploid number is the chromosome number of common wheat, Triticum aestivum. It is believed to be derived from 3 different ancestral species, each of which had 7 chromosomes in its haploid gamete. The monoploid number is thus 7, and the haploid number is 3 into 7, which is 21. In general, n is a multiple of x. X is the monoploid number. The somatic cells in a wheat plant have 6 sets of 7 chromosomes, 3 sets from the egg and 3 sets from the sperm, which fuse to form the plant, giving a total of 42 chromosomes. As a formula for wheat, 2n equals to 6x equals to 42, so that the haploid number n is 21 and the monoploid number x is 7. The gametes of common wheat are considered to be haploid since they contain half the genetic information of the somatic cells, but they are not monoploid as they still contain 3 complete sets of chromosomes. Therefore, n equals to 3x. So you can understand how understanding monoploidy gives us an insight into how a species has developed over a period of time. The next term is transcription. A gene expresses itself, it forms proteins. But what are the steps which lead to the formation of protein? This is the first step, transcription. It involves copying a gene's DNA sequence to make an RNA molecule. Transcription is performed by enzymes called RNA polymerase, which link nucleotides to form an RNA strand. They use a DNA strand as a template. From transcription, where RNA is formed, we move on to the next step, that is translation, where with the help of mRNA and different types of RNA, ribosome forms a polypeptide chain which later folds into a protein. So what is translation? If we talk about translation in a general English word, it is the process of translating words or text from one language into another. In biology, translation is the process of translating the sequence of mRNA molecule to a sequence of amino acids during protein synthesis. This means that the language of nucleotides, that is DNA and RNA, is converting into the language of amino acids, that is proteins. The polypeptide later folds into an active protein and performs its function in the cell. The ribosome facilitates decoding by inducing the binding of complementary tRNA anticodon sequences to mRNA codons. We will be looking at these terms later on. The tRNA carries specific amino acids that are chained together into a polypeptide as the mRNA passes through and is read by the ribosome. These are some of the processes that we will be talking about in detail in this paper. The next is the transcriptome and the proteome. The transcriptome is defined as the complete set of RNA transcripts in a cell produced by the process of transcription. and hence the name transcriptome, and their quantity for a specific developmental stage of physiological condition. The study of transcriptome is called as transcriptomics. In humans and other organisms, nearly every cell contains the same genes, but different cells show different patterns of gene expression. That is, in contrast with the genome, which is characterized by its stability, the transcriptome actively changes. In fact, an organism's transcriptome varies depending on many factors, including stage of development and environmental conditions. These differences are responsible for the many different properties and behaviors of various cells and tissues, both in healthy organism and in diseased organism. By collecting and comparing transcriptomes of different types of cells, researchers can gain a deeper understanding of what constitutes a specific cell type, how that type of cell normally functions, and how changes in the normal level of gene activity may reflect or contribute to disease. In addition, transcriptomes may enable researchers to generate a comprehensive genome-wide picture of what genes are active in which cell. The next word is the proteome. Proteome is the entire set of proteins that is or can be expressed by a genome, cell, tissue, or organism at a certain time. It is a set of expressed proteins in a given type of cell or organism at a given time under defined conditions. Proteomics is the study of the proteome, and it involves understanding how proteins function and interact with one another. For instance, many proteins fold into elaborate three-dimensional structures, and some form complexes with each other to perform their functions. In addition, proteins undergo modifications which may occur either before or after translation. The proteome can be studied using a variety of techniques, for example, two-dimensional gel electrophoresis and mass spectrometry. Moving to the end, we have these last four terms. The first one of the last is the genetic code. Genetic code is the set of rules by which information encoded in genetic material, that is DNA or RNA sequences, is translated into proteins, which is amino acid sequences, by living cells. The instructions in a gene that tell the cell how to make a specific protein. A, C, G and T are the letters of the DNA code. They stand for chemicals adenine A, cytosine C, guanine G and thymine T respectively that make up the nucleotide bases of DNA. These four chemicals combine in various ways to spell out three-letter words that specify which amino acid is needed at every step in making a protein. These nucleotides are strung together in a way that the cellular machinery, the ribosome, can read them and turn them into a protein. In the genetic code, each three nucleotides in a row count as a triplet and code for a single amino acid. So each sequence of three codes for an amino acid. And proteins are made up of sometimes hundreds of amino acids. So the code that would make one protein could have hundreds, even thousands of triplets contained in it. What is cistron? Cistron is the sequence of codons containing the complete information for the synthesis of proteins. Cistron is a unit of function, and very importantly, it is not the same as the gene. So what is the difference between gene and cistron? The main difference is that a gene is a nucleotide sequence responsible for the synthesis of an RNA molecule, whereas cistron is a nucleotide sequence responsible for the synthesis of a polypeptide sequence of a functional protein. Furthermore, a gene consists of both coding sequence and regulatory sequences, while a cistron consists of only coding sequences, as can be seen from the diagram. What is recon? It is the unit of genetic recombination, the smallest section of a chromosome that is capable of recombination. The last word is the term muton. It is the unit of mutation in a gene. In fact, it is a small unit. It can be either a single base or few bases, since a mutation can also occur due to one base replacement or several bases. Therefore, the smallest muton can be a single nucleotide. When considering the sizes of cistron, recon and muton is the smallest. So this brings us to the end of glossary of fundamental molecular biology terms. However, this is not the end. We will be dealing with each and many different terms as we move on with this paper. We are going to talk about the central dogma. This is very important because we have been talking about how chromosomes or DNA pass information from one generation to next, or from one cell to another, or how that information functions within that cell. How does it do it? There is a central dogma so that nature or all the species in it can maintain themselves as such. What do you mean by the simple word dogma? It is a set of principles or a principle laid down by an authority as incontrovertibly true. The central dogma of biology states that information encoded in the DNA is transferred unidirectionally to mRNA, which then directs the synthesis of proteins. The set of instructions that enables the mRNA nucleotide sequence to be decoded into amino acids is called the genetic code, as we just saw. The central dogma was proposed by Francis Crick, who has an important role along with Watson in understanding the structure of DNA. The central dogma of molecular biology predicts that a particular sequence of amino acids, a protein, cannot be used to specify or even alter a particular sequence of nucleotides, which is the gene. When the information flows unidirectionally, the information passes from DNA to RNA to proteins, but not vice versa. Instead, information flows from nucleic acid to proteins, in that an elaborate machinery exists to translate the nucleic acid to amino acid according to the rules of the genetic code. There is no back translation machinery in any cell, in any organisms, and this is how organisms can transmit only their genes to their offsprings. They cannot transmit the information encoded in the protein to their offspring. Even though the genetic material is not entirely constant, advantageous mutations do not arise in a directed manner. The predictions of the central dogma have withstood every challenge and are likely to remain as the central organizing principles of molecular biology. We have just said that it is the unidirectional flow of information, but here we can see in the diagram about reverse transcription. This was the biggest revolution in the central dogma, and this was the discovery of retroviruses, which transcribed RNA into DNA using a special enzyme called reverse transcriptase. And this has resulted in an exception to central dogma, where DNA can form RNA through the process of transcription, and RNA can also form DNA using itself as a template by the process of reverse transcription. There are also some viruses which are so primitive that they use only RNA to proteins. So you can understand how important is molecular biology if we need to understand how information flows from one generation to the next, or how that information is used by the cell in its day-to-day functioning. We will be carrying on with the rest of the paper in the next presentation.